Smooth muscle does contain actin and myosin, and they are arranged in filaments, but they're arranged differently than they are in skeletal muscle. So let's go into how that works. So for smooth muscle, let's talk about the contraction mechanism. So first, we do have actin and myosin, but we do not have a troponin and tropomyosin system. In other words, we're not going to be able to control the contraction using troponin and tropomyosin the way we do in skeletal muscle. Remember, in skeletal muscle, we have calcium bind to troponin, which moves tropomyosin, which allows the contractile cycle to take place. Since we don't have those in smooth muscle, it's not going to be able to work the same way. So the actin and myosin are not arranged in sarcomeres. So we don't have the striations, and contraction happens in all directions rather than just end to end. So in skeletal muscle, if we looked at how one muscle cell contracted, it would go from this to this. All of the contraction is inward from the ends. In smooth muscle, we have small cells which, when they contract, kind of do that, where they contract in multiple directions at once, kind of going in all around. Think of a grape becoming a raisin. So, in order to understand how all of that works, we're going to need to understand how the actin and myosin are arranged. So, let's imagine a large smooth muscle here. Now, first thing I need you to imagine are collections of protein in the cell membrane, sort of scattered around the cell. These are all in the outer membrane of the cell. So think of them as like little spots on the surface of a potato. And stretching between, actually, first let me name these. These are what are known as dense bodies. They are collections of structural proteins in the outer membrane of the cell. Stretching between them, we have lengths of actin filament. So let's sketch some of these in here. So here's one going around the other side of the cell. Here's another one. So it's almost as if just on the inside of the cell membrane we have these proteins sticking out from the cell membrane, and then there are ribbons of thin filament going from one to the other, all around the inside of the cell. So if you imagine you're sitting inside a smooth muscle cell looking around at the membrane, you'll see these dots, which are the dense bodies, and then from one dot to the other going along the inside of the membrane, not across the cell, but along the membrane, are these ropes of actin-thin filaments. They're not quite the same as the thin filaments in skeletal muscle because they don't have the troponin and tropomyosin around them. And if you look where they overlap, you will find short, thick filaments. We'll sketch those in here. So there's all of these thick filaments lying between the overlapping thin filaments. These are myosin, although it's a little bit different than skeletal muscle myosin. Now, if we take a close-up look at one of these intersection points, so let's pick this one. So we'll take a close-up look at this. Let's see, where will I put it? I'll put it over here. What you find is our thin filament here and here. And between them, the thick filament, with its little myosin heads, looks somewhat different than it did in the skeletal muscle. Remember, in skeletal muscle, a thick filament looked like a double-ended toothbrush. Myosin heads on both ends oriented so that they would pull toward the middle of the thin filament. In smooth muscle, the thick filament is shorter, and the heads on one side are all arranged in one direction, and the heads on the other side are arranged in the other. So if I think about this thick filament that I've drawn here, when those heads start pulling, the ones on top are going to be pulling the thin filament that way, and the ones on the bottom are going to be pulling the thin filament that way. So when this thick filament is working, it's going to be pulling those two dense bodies on either side of the thin filament toward each other. So when this one contracts, it's going to pull that dense body that way and that one that way. Likewise, this one's going to pull like so. So when each of these thick filaments starts working, it's going to try to pull all of those dense bodies closer together to each other, which is going to pull the entire thing inward, which is why the cell, oops, that's not what I meant, which is why the smooth muscle cell does what's shown up here, where we go from a long, skinny, spindle-shaped cell to kind of a squidgy, raisin-looking structure, because it's contracting all around in all directions. Now, there are a couple of other things about this that are important to know. So, smooth muscle, first of all, it can contract further than skeletal muscle. By which I mean, when skeletal muscle contracts, it can only go until those Z discs come up against the edge of the thick filament. So it can only contract over so much distance. Smooth muscle can work over a much greater distance. It contracts further than skeletal muscle does. And if you look back at our previous diagrams, you can probably see why. That there's no Z disc for these thick filaments to run into. So as they move those thin filaments, they can keep doing it until they reach the ends of those thin filaments. So there's a much wider range of contraction. The second thing is that they maintain strength over a wider range of stretch. Remember that in skeletal muscle, if we stretched the sarcomeres out, the thick filaments started losing contact with the thin filaments, which meant there weren't as many myosin heads to grab onto thin filaments, which meant they weren't as strong when they twitched. Again, if you look back at our smooth muscle, there's this thick filament will keep contact with that thin filament over most of its length. It can just work its way along that thin filament, and at all points, there's going to be plenty of myosin heads in contact. So when smooth muscle gets stretched, it maintains its strength over a much wider range of stretch. So those characteristics are explained by that arrangement of myosin and actin, as is the direction of the contraction, that it kind of goes in all directions all the way in at once. So, now that we understand that, we need to talk about what it is that's going to control this contraction. We've explained some of the characteristics, but not how we turn it on and off if we don't have troponin and tropomyosin. So, control of smooth muscle does involve calcium, but it's then going to involve a couple of other things, a protein called calmodulin, and two other proteins, myosin light chain kinase, known as MLCK, and myosin light chain phosphatase, known as MLCP. Those are going to be the things involved in controlling this. Now, to understand that, we have to understand one more thing about the smooth muscle myosin. So, in smooth muscle, our myosin head is a little bit different. We still have our actin binding region. The ATPase, the active site on that ATPase is normally, though, not quite the right shape. So we'll call that inactive. And we have a regulatory site here, which accepts a phosphate. This is on a part of the myosin protein called the light chain. The myosin protein is actually made of 4 smaller pieces, 2 heavy chains and 2 light chains. This regulatory site happens to be on the light chain. So the myosin is normally unable to use ATP. The active site on the ATPase is misshapen. So in order to fix that, we need to bring in a phosphate and add it. So remember, things that add phosphates are called kinases. So myosin light chain kinase adds a phosphate here, which opens up the ATPase. Once that's done, the myosin is capable of working. But before that's done, it can't work. So in order for myosin to function in smooth muscle, myosin light chain kinase has to add a phosphate to the myosin head. That takes a moment. That's a chemical reaction. We have to take a phosphate and covalently bind it to that head. But once it's on there, it'll stay. Now, sometimes, though, you want the myosin to turn off, which means we need to remove it, which is the job of myosin light chain phosphatase. Phosphatases remove phosphates from things. So if the myosin has a phosphate on it and you allow myosin light chain phosphatase to get to it, then it will remove the phosphate, the ATPase will become inactive, and the myosin will stop working. So in smooth muscle, that's how we're going to control it, by turning the myosin on and off with a phosphate. So then the next question is, how do I know which enzyme is working? Is it the kinase that's working or the phosphatase? Which one should I be looking at? And the answer is both. So let me add another thing here. So now we're going to talk about control of contraction. So let's imagine starting with the smooth muscle at rest. In that case, the myosin phosphatase, MLCP, is more active than the MLCK. It's probably about 3 times more active. Which means that while both of them are working, the phosphatase is going around trying to add phosphates to myosin. Sorry, the kinase is going around trying to add phosphates to the myosin. The phosphatase is going around removing them faster than the kinase can add them, which means most of the time, all of the myosin heads are going to be unphosphorylated. So the phosphatase is removing phosphates faster than the kinase can add them. Meaning that most myosin is unphosphorylated and inactive, which means the muscle is not contracting. Since the myosin can't work, it's not doing anything. However, when we get a signal to contract, we're going to change that. Now, what that signal is, we're going to get to that in a moment here. But so, when calcium gets into the sarcoplasm, calcium binds to a protein called calmodulin and activates it. Activated calmodulin, activates the myosin kinase. It acts as an allosteric regulator for myosin kinase, making it much more effective. In fact, it makes it work better than the phosphatase. Probably about twice as well. Which means once that happens, now the kinase can add phosphates faster than the phosphatase can remove them. So MLCK now adds phosphate to myosin faster than the phosphatase can remove it. Which means myosin becomes phosphorylated and active. And the muscle contracts. If you take the calcium away, that deactivates the calmodulin, which reduces the kinase's activity, which means the phosphatase can catch up and remove the phosphatase. phosphates. Once you do that, the myosin is inactive and the cell relaxes. So, this is a more complicated control system than skeletal muscle had. In skeletal muscle, it was relatively simple. We release the calcium, binds to troponin, moves tropomyosin, and the myosin can get to work. That's quick. Here, it's complex and slow. Here, calcium gets in. Where'd the calcium come from? We'll get to that. But calcium gets into the sarcoplasm, it binds to calmodulin, which then has to find the kinase and bind to that and make it more active, so now it catches up with the phosphatase and then starts adding phosphates to the myosin, and then the myosin's active, and then contraction can happen. And to stop it, we take the calcium away, so it comes off the calmodulin, which now comes off of the kinase, so it's not active, so now the phosphatase can catch up and it removes the phosphates. Much slower process. Which is why the twitch speed in smooth muscle is so much slower. We're very slow to build up force and very slow to take it off because we've got all this complex stuff going on involved in making the contraction happen or not happen. Now, more than most other things in the body, this is an oversimplification. Smooth muscle is complicated in terms of how it's controlled, and there's a lot of it we still don't understand. For example, we don't know how that latching mechanism works. Presumably, it involves something about the myosin. Once it gets turned off, it doesn't let go of the thin filaments, so they stay in their contracted form, but we don't know what controls that, why it works, how it works. We know there are other things that affect all this that we don't fully understand. So this is a simplified version, but it's good enough for us for now. All right, so now you understand something about how the contraction works and how it's controlled. For our final thing, let's talk about where the calcium is coming from. So let's imagine a smooth muscle cell. Now, these don't have T-tubules like skeletal muscle does. They do have sarcoplasmic reticulum. We'll just abbreviate that SR. Sarcoplasmic reticulum. And it does store calcium. And we do have gated calcium channels. These are ligand gated. And like in skeletal muscle, we do have calcium ATPases on the sarcoplasmic reticulum, which pump calcium from the sarcoplasm into that storage. So while those calcium channels aren't the same as ryanodine receptors, they're still a calcium channel. One thing we have here that we don't have in skeletal muscle, we also have calcium ATPases on the outer membrane, which means they also pump calcium out here. So we have calcium outside the cell in addition to the internal storage, which is going to give us some more options for how we can get this to work. Now, over here you can imagine calmodulin, which is waiting for the calcium. Calmodulin will bind to the myosin light chain kinase, which when it's active will start phosphorylating the myosin. So this is all the contraction machinery over here ready to go. So our question is, how are we going to get, what's going to control whether calcium shows up in the sarcoplasm? These pumps make sure it's not normally there. Normally it's either in storage or outside the cell, actually both. So how can we do this? Well, let's see. One option is actually G protein coupled receptors connected to phospholipase C. So, if you have the right ligand out here, that can bind to the G protein coupled receptor, which uses its G protein to activate phospholipase C, which makes second messengers, DAG and IP3. And IP3 in particular is a ligand for these calcium channels on the sarcoplasmic reticulum. So that allows calcium to come out. Calcium will bind to calmodulin, which will activate the kinase, which will get the muscle cell to contract. So here's one mechanism that works for this. Some things, often this can be hormones or some other messengers. Can affect G protein coupled receptors, which affect phospholipase C, which makes IP3, which regulates whether those channels are open. And there are examples of messengers which turn on the G protein coupled receptors, so they cause calcium release, and others that turn it off, so they stop the calcium release. But either way, they change it. So that's one way that a signal, like a hormone or a messenger, could cause smooth muscle cell to change whether it's contracting or not. Here are some others. You could have just a simple ligand-gated calcium channel. In addition to G protein coupled receptors. So, these are two ways that various other chemicals could cause the cell to contract. One works by releasing calcium from internal storage. The other works by letting calcium in from outside. Either way, calcium gets to calmodulin. Some other ways this can work. We could imagine a voltage-gated channel. You could imagine a voltage-gated calcium channel that when there's a change in membrane potential, lets calcium in, so it can bind to calmodulin. So this is just another way that the cell might be able to respond. It might have these channels, so that, say, an action potential in the cell would cause the entry of calcium. You could even imagine a mechanically gated channel. This is meant to be a stick. The idea being, if you poke the cell with a stick, it can open mechanically gated channels, like stretch-sensitive channels, which can let calcium in, which then can bind to the calmodulin. All of these are ways that you might be able to get a smooth muscle cell to respond. Not every smooth muscle cell will have all of these. But if it has any one of these, that's a kind of stimulus it can respond to. So if a cell has, say, G protein coupled epinephrine receptors, then epinephrine can cause a change in whether calcium is being released from inside in the cell, which will cause it to contract or not. If it has mechanically gated calcium channels on its surface, then physical force, like stretching the cell, might cause it to let calcium in and change whether it's contracting. If it has voltage-gated channels, then it can respond to action potentials. If it has ligand-gated channels, it can respond to whatever is the ligand for that channel. This is one of the ways that smooth muscle cells can be so versatile. There's a lot of options for how you might get calcium into the sarcoplasm. And now you know that if you get calcium into the sarcoplasm, you activate the calmodulin, which activates the kinase, which phosphorylates the myosin, which causes the cell to contract. So, that's a summary of smooth muscle. You may have gotten the impression, wow, smooth muscle is complicated, and you are correct. Smooth muscle is in many ways more complicated than skeletal. And that is the end of the muscular system. Yay! Good job. So, your task now is going to be to show me that you understand all this, and then we're going to go on to the cardiovascular system, where we're going to start by looking at how cardiac muscle works. And in some ways, it's a little bit like skeletal and a little bit like smooth. It's almost sort of a hybrid kind of muscle. It has a different kind of action potential. It mostly uses intrinsic control. Its mechanism is similar to skeletal, but not quite identical. So I actually think cardiac muscle is pretty fun to learn about. You'll learn about that, and then you'll learn all about the heart and blood flow. So, hope you enjoyed that. Hope you enjoyed the muscular system. See you for the cardiovascular.